Okay, so I think everyone knows Felix Ritort. He's a CREA professor in the department, and also he's the head of the Smart Biosystems Lab, which is a very successful lab also in the department. And he agreed to give us a talk showing the latest things that they've been doing there. So Felix, please.
Okay, so thank you very much for the kind invitation. It's a pleasure for me to present a review of the things that we are doing in my lab. Many of you probably already know it, but I think there are some other new things that for those of you that just enter into the world of the optical traps, maybe it's useful. So what I have done is I will present a brief introduction to optical trapping, and I show a bit a few selected applications on on some of the questions that we are working in my lab, in particular information to energy conversion. So let me just start by explaining very briefly or mentioning the principle of optical trapping. Optical trapping is based on the idea that light carries momentum, and therefore, upon deflection of a light beam, you can exert forces on small particles. And here in the slide, you see a schematic representation of a bead on the size of a micron, okay, microns typically, embedded in water. Water is represented by the yellow background, in such a way that the light ray that is colliding or is colliding against the particle is deflected just because the index of refraction of the bead it's bigger than the index of refraction of water, and therefore there is, according to the Snell's law, a change in the light direction. Therefore, at the end of this deflection, upon crossing the bead, the outcoming ray changes its light momentum because it changes the direction of motion, the direction of the photons, and therefore there is a net change of light momentum that means that or implies that there is a reactive force shown by this blue vector that opposes this change in light momentum, and therefore light exerts what we call optical forces, and in particular gradient forces on particles. Okay, so this force that is exerted on a particle is used for optical trapping if you use a microscope lens, an objective that has sufficiently high numerical aperture, which means a sufficiently high ratio between the diameter of the lens and the focal length, and a laser beam that is focused in a spot inside water. In this case, that's the same condition as before. We have a bead in water, and if we focus this incoming laser beam in a point, then there is an attractive force, a net attractive force, acting on the bead relative to its equilibrium position. And actually what happens, if you see me in the screen, is that in the focus of light, we can use the focus of light, the focus point, the focal point as a force, as a point in which there is a spring acting on the bead, and this is acting like a force sensor. So essentially, if you measure the deviation of the bead with respect to the focal point, or the equilibrium position, we can use this deviation, for example, by video microscopy, to report and to calculate the force acting on the bead. So it's a very useful means of measuring very tiny forces. It turns out that the light momentum is very low, and the forces exerted by this deflection of the light beams are very low. Altogether, the typical forces that we can measure are in the range of piconewtons. And this piconewtons is what makes it interesting, because it can be applied for many biophysical applications. Now, this optical trapping principle, okay, was discovered, let's say, by Arthur Ashkin in the 70s, while he was in Bell Labs, and this led him to this. He was awarded for this discovery the Nobel Prize 2 years ago. Okay, and this was shared with other people. He got half of the prize in 2018 and was shared with Gerard Mourou and Donna Strickland, who got the other half for applications of lasers, okay, in solid-state physics, applications of lasers in the high power, very high power regime, okay? And I had the great opportunity to attend the Nobel celebration that year in Stockholm because, well, I contributed as reported for the Nobel Prize, for the prize that was awarded to Arthur Ashkin in recognition to that contribution. So they invited me. That was a very interesting experience. Now, what I want to show you is the principle of optical trapping, as you saw before, which is a lens with a high numerical aperture, now has become more sophisticated in the form of instruments that combine more than one optical trap. And in the instrument that we have in Barcelona, we have several instruments of this kind. We combine two optical traps that we position in the same point to obtain a more strong optical trap. And for reasons that are not essential, but for practical applications, it turns out that using two counterpropagating traps, two counterpropagating beams, like you see here in this representation, that focus on a spot, lead to a much higher optical trapping force, which at the end is the gradient force. And what you want is that the optical gradient force, which is this force here, which goes in the optical axis direction, is much higher than the so-called scattering force, which is a force that is, if you want, a consequence of the light pressure exerted by the beam on the bead. So you want to have a very stable optical trap. You want a high optical gradient force, and therefore we use the counterpropagating beams. I don't know if you are seeing my cursor here on the slide. Do you see it, or?
Yeah.
Okay, that's good. Now that's the instrument in a compact form that we have in the lab. So it's everything very compact, and these reduce considerably the drift in the measurements, which is something that on molecular scale is very important. Okay, so we have a resolution of about 5 angstroms. That's the resolution in measurement of the position of the instrument, half a nanometer, and of course in this scale drift is very important. So the more compact you do your instrument, the better it is, because you reduce sources of noise like dilatation parts of the instrument, or more important, the optical path, the length of the full optical path of the light beam in the instrument. So a remarkable experiment that shows you the power of this technique is what I write here. You pull a single DNA molecule by attaching the ends to two beads, then you move one bead relative to the other bead, and you pull the molecule, you exert a force and measure the force extension curve. So pulling a single DNA molecule, you can imagine now it's quite a standard in many labs. It's not an easy experiment because you have to prepare the molecule. There is a biological synthesis part that you have to learn about how to do it, but essentially, if you think about it, it's a remarkable thing because it's testing, if you want, the molecular hypothesis. You can test the molecular hypothesis at its best because you pull a single molecule, so you probe the existence of single molecules, which of course they are not single atoms, they are single molecules, but of course these are polymers, so these can be thousands or hundreds of nucleotides after each other. So it's a big molecule, it's a polymer, right? But you can see it in real time, and that's makes it remarkable. So the idea of exerting force on molecules is not, it dates back to, I would say, 30 years ago in 1990, when Carlos Bustamante, at that time he was in Oregon, thought about the possibility of using gravity as a force to pull on individual DNA molecules. So he used a cover slip to which he attached on its upper side DNA tethers, and at the end of this tether there were beads. Okay, now the beads were used to measure the height of the DNA molecule, the length of the stretching of the DNA molecule, by using video microscopy. And by using gravity, he was able to measure 3-4 points in a force extension curve. So by changing the density of the beads, he was able to control, let's say, the Archimedes, the sinking force, the sedimentation force, okay, of this bead in water, okay, and changing the size and changing, but especially changing the size, changing the density, you could exert different forces, and therefore you could put a few points in this plot of force versus extension, okay? And he found this points here. He tried to fit to a freely jointed chain model, which was a model used at that time from polymer theory, and the freely jointed chain model predicts a linear behavior of force versus extension that afterwards bends up due to the entropic elasticity. And well, he found values of the persistent length of 500 angstroms. Okay, this is a curve which is close to the real value, because the value of the persistent length in deep double-stranded DNA is about 50 nanometers. Now, these were very primitive measurements. The idea came then much better with first some experiments with magnetic tweezers in 1992. And then in 1995, there were these, let's say, definitive experiments using optical tweezers in which they could it's possible to get full force extension measurements along a full curve, not just for 3 points. Here you see a setup. You take a DNA molecule, you label at the ends, and you attach the molecule to 2 beads. One bead goes immobilized on the tip of a pipette. The other bead is captured in an optical trap, and this is the bead, okay, that gives us the force measurement. So the measurement of the force that is applied at the end of this DNA molecule through this biotin-streptavidin contact. Okay, this is a near covalent bond which is capable of sustains forces or can resist forces up to hundreds of piconewton. This is less effective. This is an antibody-antigen interaction. It's called deoxygenin anti-deoxygenin binding. It's much weaker, but overall, by putting several deoxygenins, we can reach up to forces that easily go to 100 piconewtons. And here I show you a video of the experiment in which we immobilize the 2 beads. We trap the 2 beads. One is immobilized on the tip of a pipette. The other bead is in the optical trap and has been coated with DNA molecules. So it has DNA molecules on the surface, and therefore we can set the pulling assay. We can set the experiment and pull the DNA molecule while we measure the force and extension. So here you see we use the optical trap to capture a bead. We are moving the chamber with motors. We immobilize on the tip of the pipette by air suction. All these we build in our lab. All these parts, all these materials, we make them in our lab, the pipette and so on. And here is another bead. This is another bead which is bigger. It's 3 microns diameter. This has been incubated with DNA and has DNA absorbed on the surface. And now we position the big bead with DNA on top of the small bead. And now the idea is that we have to fish a DNA end from the big bead in the optical trap. So we start moving the pipette, but let me just stop one moment here. As you see, here we are pulling a molecule which is called half lambda. Lambda DNA is a DNA that comes from the bacteriophage of a virus. Okay, is a bacteriophage is a bacteriophage virus from a bacteria. Okay, and this not only humans have virus, but also bacteria suffer a lot from virus, and they are attacked continuously by virus. And these are called bacteriophages. Okay, so bacteriophages are abundant in nature, and there is one bacteriophage which is called Lambda virus. And Lambda virus was discovered in the 40s in the guts of Paris, and was kept in the fridge by the phage group, Max Delbruck and all the people who preceded the discovery of the DNA structure, which was by Watson and Crick in 1953. So they kept in the fridge as this Lambda virus, and now it's everywhere in all labs, okay? But if you find it in nature, it's not anymore in nature because it has mutated and has disappeared. But we use experimentally this Lambda DNA from the virus in most of our experiments. So now this Lambda virus that we are pulling here is 8 microns long. So you see here in my bar, these 10 microns is longer than the 8 microns. So what happens is that DNA is not extending here. The macroscopic idea in physics, in polymer physics, is that you would have maybe something which is long, but that's not true. Polymers form a random coil due to the thermal forces of the bath. And therefore, these 8 microns of DNA are not protruding outside from the bead, but on the surface, like some sort of brownies, okay? You could call, or bagels, okay? Or ensaimadas. So they are on the surface. So if I want to catch the other end of the DNA, I have to approach this bit to the surface if I want to catch it. Unless I put a flow. If I put a flow, I could extend the DNA molecule and could catch the end. This is another way, another method of capturing it. But here I show you the first one in which it's forming a random coil on the surface, and therefore I have to approach the pipette, the bit in the pipette, the small bit to the surface. Now you see that I will approach it now, and when I approach it to the surface, I will catch the end of the polymer, which is a random coil. Now I catch it. It's there. I don't touch the other bit, but I catch it. And now I have it, the connection. Now I can start measuring because I have a tether, and I measure the deflection of the force by measuring the change in light momentum using position-sensitive detectors in my instrument, while I measure the extension of the molecule. With I could do video imaging here. We use another method, but it's basically equivalent. Okay. And this is what you get for the force versus extension of this DNA molecule. That's force in piconewtons. It goes from 0 piconewtons to 80 piconewtons, okay, or a bit higher than 80 piconewtons. Here is extension. The molecular extension is in nanometers. It goes up to approximately 16 microns, okay? But you see that vertical dashed line is the contour length of the DNA of this virus, which is this half lambda, is half of the lambda, okay? It's about 8 microns. So the molecule shows a response, which is this entropic elasticity at low forces, below 5 piconewtons typically. You have the entropic elasticity, but then at some point you overcome the contour length. You go farther. How is this possible? Because the molecule is 8 microns, but if you stretch it, you get a longer extension. And then you reach, so you overcome the contour length, and you get this plateau where the molecule overstretches by 70% of its contour length. And it's thought that mostly this is due because DNA is a double helix, and you will stretch to very high forces, then it uncoils. And by uncoiling, okay, you get a further extension. You go from a helix to a ladder, to a tilted ladder, okay? So that's the structure of DNA. So as you know, DNA is a helix, but for which reason it's a helix? In principle, it should be a ladder. But we know that bases are hydrophobic. They have London forces. P stacking is called in chemistry, which makes this become a double helix. So you will see this transition here. And you can fit this low force regime. You see here an inset in this square from 0 to nearly 20 piconewtons. We fit with a warm light chain model, which is a version, more elaborated version of the freely jointed chain models. And you see this is the red line, the model. The green line, there are two green lines. There is the noisy green line, which is the 1 kilohertz measurement with thermal fluctuations, Brownian fluctuations. If we filter out the signal to 10 hertz, okay, then you get this line, sorry, to 2 hertz, you filter to 2 hertz, okay, then you get this green line, which is continuous, which is perfectly fitted by theory. So the theory, the red, fits the green perfectly, especially in this regime, okay, where you have entropic elasticity. So you get this value, you get these values of the persistent length, 45 nanometers, and the Young modulus, which is this further lengthening of the contour length. So this is like, this Young modulus is also called the stretching modulus, okay, and is in this case 1,000 piconewtons, which means that every 10 piconewtons, the contour length is extended by 1%. Basically, it says that every 10 piconewtons. So that's the best numbers now people can get for these elastic parameters of DNA, and this is possible with these force techniques. Okay. So the applications of force spectroscopy and optical tweezers are large. Here I listed a few examples to molecular thermodynamics and kinetics, nucleic acids, proteins, sugar polymers, lipids. It can be applied to any sort of relevant biomolecule. It can be applied also to investigate molecular machines, okay, the transaction of energy from ATP or GTP hydrolysis into mechanical motion. And this is a very successful line of research, which many people throughout the world is using optical tweezers for that. You can also use it to investigate intermolecular bindings, for example, how proteins bind to nucleic acids like DNA, RNA, how peptides, for example, bind to DNA and intercalate. And this is, for example, important for the research, cancer research at the level of understanding how drugs attack or how drugs interact with nucleic acids and prevent replication of the DNA of cancer cells. So that's an important application in biomedicine, I would say. There are also important applications to measure, for example, antigen-antibody affinities. So, for example, you could apply it to know what is the strength of the interaction between the coronavirus, for example, and a human cell. You could do that. Of course, these are hard experiments, but people are working on this now, right now, okay, because we had this year of the pandemic. So these applications are going on, and you can apply it beyond molecular interactions also to cell biophysics. So you can do what we call cell phenotyping, which is characterizing cells by their mechanical properties. For example, to understand whether the stiffness of a cell gives you information about its function, about its internal state, and other applications. And also there's optical tweezers, and we were talking about this with Antonio Ortiz, who is organizing this meeting, this Skype meeting, but this conference online, let's say. And with Antonio, we were talking about optical traps can be used also for fundamentals to understand, to validate, to test predictions from theories in non-equilibrium phenomena, okay, non-equilibrium physics in general. And also I want to mention this application information theory. We can also use these force techniques not just to measure energies, okay, in joules, in kcal per mole. We can measure beats. So that's quite remarkable that you could use force measurements to measure beats, okay, and that's something we can do, as I will show you later on. So about force spectroscopy and applied to molecular thermodynamics, I collect here one or two examples. Basically, what you can do is you can apply force to any molecule and break molecular interactions. For example, you can unzip DNA by pulling apart the two complementary strands in a total helix. You can also unzip RNA that is forming a structure, a folded structure, okay? But here is a double-stranded nucleic acid. Here is a single-stranded nucleic acid. So you pull from the 5', 3' end of a nucleic acid, in this case an RNA, and you can reveal its molecular structure, okay? I will show you an example later of this. Also, you can pull proteins, okay? You can pull from the N and C termini, and you can, for example, investigate how proteins fold to the native state. That's something you can do with the technique. Here I show you the example of unzipping. So here you see that's the unzipping experiment. We take the double-stranded DNA, we pull from the ends, and we measure this curve here. So this is unfolding. This is unzipping of a molecule which is about 7,000 base pairs. Every base pair contributes to about 1 nanometer of extension, releasing extension. So this curve, the full unzipping of this DNA molecule, which is 7,000 base pairs, goes up to 7 microns, 7,000 nanometers, which is here. Okay, we'll stop now. And you will see that at the end, I have a hairpin because I want to separate the two strands, but keep the same molecule intact in such a way that I can reconstitute it when I release the force, because I want to repeat this experiment many, many times. So what can I do with this experiment? With the experiment, because I'm breaking the intermolecular, I'm breaking the base pair interactions between the two complementary strands, I can measure the energies of hybridization of the base pairs. So I can extract experimental measurements, I can extract numbers for the energy of forming GC and AT base pairs. And this is very important for DNA thermodynamics, to characterize the energy that makes a G to bind a C or an A to bind a T, okay? And as you see, this is the 7,000 microns molecule, and here is the full pattern of unzipping. This depends strongly on the sequence of this molecule. If you change the sequence, you get a different pattern. So this is noise for sure, but on top of noise, there is sequence information, there is signal, okay? So you will see it now, because when I release the force, I come back, this is a single-stranded DNA response. When I come back, which is the blue curve, which I stop right now, you will see that I reconstitute the same pattern, okay, because it is based on the sequence. So this could be used for DNA sequencing, actually, okay? Now you see that the force is typically 14 piconewtons. This is the typical force to break GC and AT base pairs, okay? So let me show you how this remodels and reconstitutes again to form the same 7,000 base pairs hairpin, and you will see that it's nearly reversible. That's one of the most important messages of these experiments, is that biological matter is reversible, is out of equilibrium, but has this propensity to reanneal and to reform again. Take any material, any solid material, and break it, and then try to reconstitute it. You will never get it. But with biological matter, this is the great and marvelous feature it has. It can remodel, and the information can be recovered back again. Okay, so this is a fracture reversible, not irreversible, is a reversible fracture problem. Okay, that's very special of biological matter. And you can do this and also investigate, for example, I don't want to go into many details, but how, for example, we investigated in my lab a few years ago, how peptide intercalates DNA. This is a drug that is used for as an anti-cancer peptide for mammalian type cancer, for breast cancer, for breast cancer. And well, it's not commercial. It didn't reach the commercial step, but it was it went up to the clinical trial number 2. Okay, so many of the drugs that are in biomedicine do not reach the level of being sold to people. They have to pass very stringent levels, tests, trials. So there is trial level 1, trial level 2, up to the final one, and most of them stop in the first 2 levels, let's say. But you can see that the intercalation of the peptide reveals a series of force rips at very high forces, which go up to 25 piconewtons. And the reason is that a bis intercalator is a molecule with 2 moieties, and the 2 moieties enter into the double-stranded helix, and therefore block it like a staple. You know, a staple that you put in a paper, and it fixes the distance between consecutive bases and does not allow to unzip the molecule. So we can see this as these big, large force steps. We have used these measurements in this case of this experiment of unzipping that I show you here also again. Here is the raw data, here is filtered. The 2 colors are unzipping and rezipping, so you see it's nearly reversible. Okay, the black is unzipping and the red is rezipping. There is nearly no hysteresis. They collapse on top of each other. Therefore, this is an equilibrium equation of a state. It's an equation of a state of our system in equilibrium. Therefore, we can use that to extract the free energies of all base pairs in DNA. And we did that a few years ago. We published a series of papers on this, in which we measure for 2 different molecules of different length, 2,000 base pairs, 7,000 base pairs, and we found the same. We compared to measures obtained in calorimetry, which is measurements that are used by servers of native structure of nucleic acids predictions. So for example, one of them, the most well-known, is M-fold. There are others like the Vienna package. So we compared these and we got a good agreement, okay? There have been other developments I want to mention, this sort of review. For example, optical trapping has been used in 1993 for the first time by Steve Block in Stanford, where he was able to resolve the steps of kinesin. And he saw that the kinesin does the steps by 8 nanometers. Now there have been this story of the kinesin walking along a microtubule has had a long story. The most recent paper, it has appeared in Science, if I remember, just a few months ago, by Eric Schaffer. Eric Schaffer has done a beautiful paper. I recommend you to look at it. Jaume, you should look at it. It's a beautiful paper in which he uses germanium bits to reach very high efficiencies. And he has been able to track the true motion of the kinesin that works in a hand-by-hand way. This has been proposed in the past as a scenario, but he has done the experimental validation of this hypothesis. A very beautiful paper. Okay, so he uses metallic beads to get a higher signal for optical trapping rather than plastic beads that, for example, we use in our lab. Other applications are to non-equilibrium physics. I show here 2 papers or 2 contributions, let's say, of things that I like. One which I like, which I think is great, is really very nice, is this work by the group of Michael Woodside. He's in Alberta. Michael Woodside has been able to see how the molecule that can be folded and unfolded, how an individual molecule, which has 2 conformational states, jumps from one to another by jumping over a barrier. He has been able to see the diffusive motion across the barrier. So here you have what we call the reaction coordinate of this molecule, which is its molecular extension, which jumps between 2 values, between 2 levels. And you see that the molecule is folded and then jumps to the unfolded. Typically, when you see a signal, if you look at the millisecond timescale, what you see is this. You see nothing. You see basically a single jump. It's a dichotomic signal, okay, where you see a jump between 2 levels of extension in this case, but you don't see structure in these jumps. Now, if you go to the microsecond timescale, which is shown here by this time bar, you can see that this discontinuous jump in the second timescale, in the millisecond timescale, when you go to the microsecond timescale, you see that it's not really a jump, it's a motion which is diffusive. So this is the typical, this is the prediction by Kramer's theory. So crossing a barrier, a kinetic barrier, is not a process that is just advective. It has a diffusive component. And you see here, because the extension goes back and forth many times in the barrier, in the top of the barrier. This is a remarkable feat. And also I want to mention something which I have worked, which is this Jarzynski equality, or the possibility to extract free energy differences from reversible work measurements, okay? So this is very important, because this teaches us that in small systems, you can measure free energy difference by measuring the reversible work. And this goes against any thermodynamic textbook that you can read up to the year 2010, possibly, in which they tell you that free energy differences can be measured only by reversible processes. You won't find any book telling you that you can do that from irreversible processes. But the Jarzynski equality, that's the main contribution, says that it's possible indeed in a small system when you can measure fluctuations. This has been tested experimentally first in this paper in 2002, and then later myself in 2005, I did an extension of this Jarzynski equality to the Crookes fluctuation theorem, where we could measure the free energy as well. Okay. In my lab, we have been working on this for several years, measuring delta G from irreversible experiments using either the Crookes fluctuation theorem or its corollary, which is the Jarzynski equality. And we have published a few papers on this, from, let's say, the free energy of native structures to the free energy of kinetic structures. We can measure now the free energy of formation of misfolded states, of intermediates. And not only that, we can also apply this to measure the free energy of intermolecular binding. We can measure affinities. We can experimentally verify the law of mass action. We can do beautiful things. The message is that whenever you can measure work, you can always extract thermodynamics. And we are now applying this. We have applied this recently beyond the reversible case of unzipping to the irreversible case of unzipping, because RNA, a difference with DNA, has an irreversible response when you do experiments. You cannot do the experiments that I showed you before with DNA in equilibrium for the case of RNA. RNA is much more irreversible because RNA has a much complex structure. So when you pull it, you see reversibility. So what we have been able to do is, in order to extract the thermodynamics of RNA, we have been able to derive the equilibrium curve by the assembling piece by piece of all irreversible regions by doing many pulling forward and reverse experiments. So that's the most, let's say, if you want, is the most complex application of this sort of fluctuation theorem relations to measure free energies, in which we can have even 6 states. So we can measure the energy of each 6 states, intermediates, okay? Another application, always in the field of molecular biophysics, is that we can measure stabilization energies of magnesium ions to RNA. This is RNA of a ribosome. Well, this is a whole ribosome. So here you have protein and RNA. As you know, the ribosome is a huge molecular machine. is the biggest molecular machine in the cell, okay, in which you have protein and RNA. And this is the last X-ray image of the ribosome by cryo-electron microscopy, not X-ray, sorry, by cryo-electron microscopy. It has been published like 1 or 2 years ago, this beautiful structure, which is the highest resolution structure we have at this moment. Now, it turns out that this is stabilized by magnesium ions. This structure, you see these green balls here everywhere, are magnesium ions. Magnesium ions are essential to form these three-dimensional structures. But the measurement of the magnesium binding energy is very difficult and cannot be done. There are very, very few methods and are very indirect. But with optical tweezers, we can do that. And here I show you one example of one substructure. Is a part, you know, is a part of this big mess. Is one part. There is a three-way junction here, which has several magnesium ions that bind to this structure. So we isolated this structure from the whole ribosome and then measured the binding energy of magnesium. There are 3 magnesiums that stabilize the junction, and we use these fluctuation theorems to measure the magnesium binding energy. And we got this measurement, which cannot be obtained otherwise, unless you do numerical simulations and other also sophisticated approaches. So that's a very good way to measure the specific binding energy of magnesium ions. So that's the last part of my talk, and in which I want to show you how we can use single molecule research not to measure energies, but to measure information. And here I like to always to show this slide about measuring information. What is information? Of course, there is information theory, and you know about it, and it's very widespread in science and now in physics also, of course, especially in quantum mechanics. Information theory is very important. Okay, but now it turns out in biology is also very important, biophysics, and in physics itself, in statistical mechanics. So how can we understand information? And my answer is by measuring it. If you measure something, you start understanding it before you read anything in any book. Okay, so that's I am advocating for this statement of Lord Kelvin, who said more than 100 years ago, you start understanding something when you can measure it. Something that you don't know how to measure it is hard that you understand, because you cannot explain it to anyone how you would perform actually, how could you access that measurement, right? So single molecule research is a good playground for that. And here I show you how information goes into the statistical mechanics. Let's say, how is a concept, is a paradigmatical concept in statistical mechanics, how it's linked. And this goes back to Maxwell, who proposed that you could use a small being that in an effortless way could open and close a gate to separate fast and slow moving molecules in a initial homogeneous container to reach an inhomogeneous container where the fast molecules and the fast molecules are on the right, the slow molecules on the left, and you have a temperature gradient. So it turns out that you can do this process, and this has been so this violates the second law somehow. Why? Because this demon could, in principle, do this by operating in a frictionless gate. And therefore, ideally, this process of separating particles could be done at zero work. And therefore, as you know, with zero work, you cannot establish a temperature gradient because the total entropy decreases, and therefore it goes against the second law. So the total entropy here has decreased relative to here, and this has been done without expenditure of work. And therefore, what's the solution of this paradox? Well, this Maxwell-Demon paradox has gone for a century, basically, and it was solved, I would say, basically in the 60s with Landauer and in the 70s with Charles Bennett, who demonstrated that the statement that the entropy cannot decrease is true in a whole cycle. So you have to go back to the initial state. Now, it turns out that the demon doesn't reach the initial state again after doing this separation procedure. Why? Because The initial state of the demon is one in which he has not made any observation yet. The final state of the demon, he has accumulated some information that he has stored somewhere. So in the process of separation, when the demon saw a fast-moving molecule, he had to say, okay, this is fast-moving, it's 1, or this is slow-moving, is 0, and therefore he had to annotate something, this information somewhere, in a computer, in a notebook, in the brain, wherever. To come to the initial state, this information that has been collected from here to there has to be erased again. And when you erase information, that's the Lambda principle, you produce some energy. Whenever something remodels to reach an initial state, you expend heat, you produce heat, you expend energy. When a cell during the night you are sleeping remodels and a tissue is regenerated, you produce heat. And that's a principle of data erasure, coming back to the initial state. And therefore, this is the solution of the paradox that is accepted now, okay? But it took 100 years to reach to this solution, okay? So it was a long time until it was understood. And it has been used also, a variant of the Maxwell demon is the Szilard engine that can be used to extract heat from the bath to convert food into work. This again violates the second law. You cannot convert heat into work with 100% efficiency. Of course, you can convert work into heat with 100% efficiency, but not the reverse, okay? That's the second law. And he invented this variant of the Maxwell demon in which you could do that by taking a molecule in a vessel, in a vessel separated by 2 compartments, and depending on which compartment you are, so then you can put a wall and then extract work by using heat from the bath, okay, to lift a weight, okay? So we applied recently, we did the realization experimentally in my lab with an optical tweezer in which the role of the weight that is pulled by the molecule is played, let's say, by an optical trap. So what you do in this case is just you look at the state of your molecule that is hopping between 2 conformations, and depending on the state of the molecule, you put a wall, you move the optical trap in one direction or another. So wall insertion is equivalent to moving the optical trap instantaneously, okay? And if you do this, here is a trace, a typical trace. You look, the molecule is folded here and folded here, and with folded, you move the optical trap and then you recover again the initial position. That's the moving, that's the pulley mechanism, but exerted by an optical trap. And all these transitions are work extracting events. So what we were able to do, is to apply this and experimentally measure the work distributions at different conditions of this hopping trace of the molecule and test the formula predicted by Landauer, which is that the entropy, the maximum work you can extract is given, okay, by the Shannon information in 1 bit. So that's basically the 100% efficiency in conversion from information to energy of this machine. So here is the line, and the Landauer limit is KBT log 2. That's the maximum work you can extract. We introduce a variant of this Maxwell demon in which we did many measurements one after another, but that's something I don't want to discuss. And now just to end, because I think may have 5 minutes still, or yeah. I don't hear you.
Okay.
Yeah. So let me tell you that coming back to pulling single molecules again, you can do very interesting things like not only measuring free energies, we can also measure enthalpies and entropies. So one thing that I will show you is some experiments we have done with proteins. So let me just come back to this presentation. So when you pull a DNA hairpin, you can go from the native to the unfolded state just by increasing the force above 50 piconewtons. That's an animation of the experiment, okay? So the molecule is repeatedly folded and unfolded, okay? And this you can do with DNA hairpins, you can do this with RNA hairpins. The interesting thing is that you can also do with proteins. And with proteins, is particularly interesting because in proteins, different with this hairpin that I showed you before, proteins are strongly irreversible. So typically when you unfold a protein, the fourth rip of unfolding, because you release some extension, so the force drops, occurs about a force, but the refolding is very slow and occurs at low forces, okay? So one thing that we have been doing in my lab recently and is that we have built a new instrument that is capable not only of measuring, so it does not operate at room temperature, but it can operate at different temperatures by using what we call a heating laser. So there is an additional heating laser that allows you to reach higher temperatures. Now, what's the interest of reaching higher or temperatures? The interest of reaching higher temperatures is that you can apply the same as I showed you before to measure free energies at a given temperature, but you can extend to other temperatures. But if you know the free energy at the different temperatures, you know that entropy, okay, is the derivative of the free energy with respect to temperature. So actually, by measuring at different temperatures, we not only can measure free energies, we can measure entropies, and therefore we can measure enthalpies. And this is very important because this gives you the full picture of molecular thermodynamics. So we have investigating this protein. Not only we can, with the instrument, heat up the temperature, above room temperature, which will be 25, we can also cool down and reach up to 4 Celsius, okay? And what you can see is that by cooling is done by using a fridge. We put the instrument inside the fridge because a small instrument, you remember it was a head it was a box basically. So we put the box inside the big fridge and then we can cool down and we can operate also at different temperatures in the cold freezer mode by using the heating laser. So we can span from 4 Celsius up to nearly 40 Celsius. Okay, that's something we have done in a protein which is called Barnase. Barnase is a 110 amino acids protein, and we have been investigating the kinetics of folding as a function of temperature. I will finish here. I will just tell you that with this kind of measurements, we can investigate what is the free energy, what is the enthalpy, and the entropy of folding. We can also extract the kinetics, and we have possibly, I could say, the most complete characterization at a single molecule level of the folding of a protein, at the single molecule level. And that's something we have not yet published. We are on this right now, busy writing the manuscript, the paper. I will end up and thank the people in my group, and of course also the funding, and be very glad to answer any question you may have. Thank you very much for your attention.
Thank you, Felix. So if anyone has any questions, Pietro? You're muted.
Yeah, okay, now I'm not. Very nice talk, Felix. I already listened a couple of times, some similar talk from your group, but I find it very, the experiments that are beautiful. I have a couple of curiosity. One is related to this zipping and unzipping that appear to be totally reversible. That is a very nice experiment. So when reversibility broken, so you need, for example, 20, 30 experiments, and then you see that one of such a small some element of the chain is broken, and then you do not have the coils. So there is a threshold beyond which, on the number of cycles that you have to reach to break this reversibility. And then the other one is related to this Jastinsky inequality. Whatever there is a critical size beyond which this equality is broken, or if someone is checking. Thank you.
Okay, these are very good questions. Thank you very much, Pietro. So first, regarding reversibility, what determines the reversibility of the unzipping experiment is the pulling speed of your optical trap. Because to unzip, this is a hairpin that you are unzipping. You see my cursor. You move the bead upwards, downwards. When you move upwards, this is the black part of the curve, which is behind the red. You don't see it well, but it's behind the red. When you relax, when you retract the position of the optical trap, to reach the original position, you move downwards the optical trap, and this is a red. Now, what determines the reversibility of this curve is the speed at which you move. Okay, so if you move very fast, okay, then if you move very fast, then you would see that there is a difference between the black and the red. So these experiments were done by moving the optical trap at 20 nanometers per second, which is among the lowest speeds we can implement in our piezoelectric device. Okay. In that case, you reach a quasi-equilibrium curve because hysteresis is negligible.
So could we say that we have a adiabatic basically here? Exactly.
Adiabatic, not in the sense that the heat is zero. Adiabatic
no,
in the sense
that it's extremely slow, almost equilibrium, right?
Almost equilibrium. But you see, for example, here, Pietro, you see here, the peaks in the force. Now you say, okay, I want to do this reversibly. That's not possible because the staples, when they unbind, the time scale you should wait to reversibly rebind them on the same position, okay? It's nearly impossible because first they won't rebind to the same position. And second, these time scales can be huge. And the same happens for the RNA. The RNA is irreversible. So you tell me, oh, Felix, why don't you pull it slower? Well, the point is that if you want to make this really reversible, it's not that going to 10 nanometers per second or 1 nanometers per second will help. Possibly I should do 1 nanometer per day because you know that the time scales depend exponentially on the energies. Now, as soon as you go into something non-equilibrium by a barrier that increases 10 kVt, you are doomed. You cannot do it. Okay. Yeah. And the second question was about the Jarzynski equality. Yeah, that's correct. So this equality is based on the fact that this equality, what contributes most to this equality? So to this average, the exponential average here is dominated by the lowest work values. This means that you have to measure the tails. So if you go to a macroscopic system, and you know, your work distribution will be Gaussian because in macroscopic systems, everything is Gaussian. Yeah. But then your average work will be proportional to the Avogadro number because energy is extensive. So your average work will be proportional to whatever will be larger than the difference in free energy because that's the second law, but also will be macroscopic. So you will never catch the tails. So it will break down. So in macroscopic systems, it breaks down by definition. In small systems, it doesn't break down because in small systems, here I show you forward and reverse work distributions going in the process, let's say, going forward in one direction and then reversing the process. When you reverse the process, the distribution is somehow symmetrically related, but they overlap each other. And in this case, you are very far from the macroscopic regime. In the macroscopic regime, you would get a delta Dirac here and a delta Dirac there. So you don't get the tails. So the answer to the question is that it depends when the free energy the dissipation starts to be 10 kVts. 10 kVts, you can do it. If your dissipation is 100 kVts, forget about it. If your dissipation is 10 to the 23 kVt, then it's even worse.
Yeah.
I don't know if I answer your question. But...
Yeah, sure. But it's whenever there is a net transition from a small, a big system, the question is what is small relatively, where Jarzynski apply and so on. But basically, yeah, you answered both questions.
Yeah. Basically, I would say that Jarzynski, you can test it or it holds with the data that you have when your work distribution catches in your fluctuations of the work, you catch trajectories where the energy coincides with delta G.
Okay.
So if there are trajectories, a finite probability, if there is a finite probability, whatever, 10 to the minus 3, 10 to the minus 3, but not 0. When you have a finite probability that you catch trajectories that have a work equal delta G, then you are there.
Okay. Thank you, Felix.
Is there any other questions? Okay, so I have one maybe. So basically, and it's related to what Peter asked. So basically, what you're saying is that this Jarzynski equality holds whenever you have something that is ergodic. It's that related to the, because when you start saying that you have hills that are unsurmountable, it sounds to me.
Yeah, it's correct.
Okay.
Actually, I mean, yeah, when you are start seeing reversibility, you know, when you have trajectories that start sampling the configuration of the space, when you start seeing the red trajectories that make your system ergodic, you could say that. In fact, you know, there is the when there was a problem, when the question of irreversibility was posed more than 100 years ago with Boltzmann and others, the question was why we don't observe these trajectories that go backwards in time as we observe the trajectories that go forward in time. Why the change in entropy is, the net change in entropy is positive? Because there is the same probability. In principle, there is a probability that we observe also the reverse trajectories. And that's true. There is always a probability to observe the reverse trajectories. And there was the Loschmidt paradox, which said that if you reverse the velocity of particles at a given time, you will go back to the initial state. And that's true, because the Newton equations, if you take a given configuration and you reverse at a given time all the velocities of all particles in your system, you go back straight away to the initial state. Okay. Now, the answer was that, okay, yes, but this trajectory has a very low probability. So, of course, if you do it, if you condition your question to the fact that you can reverse the velocities, then you would do it. But this doesn't happen in nature. So this configuration where the velocities are spontaneously, all of them reversed, won't happen, never. So condition that it happens, of course, you will come back to the initial state. But what is the probability that it happens? It's zero. And this relates to the Poincaré paradox. The Poincaré paradox is that all systems, because if systems are ergodic, you would come back at the initial configuration if you wait a time that typically increases exponentially with the number of degrees of freedom. And here is the problem. For a small system, the number of degrees of freedom is small. This Poincaré recurrence time, then it's huge, but not as huge as if you put in your exponent the Avogadro number. So we come back at this idea that this is related to ergodicity. So your question is right, yeah.
Okay. So if there's no further questions.
Yeah, I have one question.
Yeah.
Well, Felix, it's just something that puzzles me about your statement that information is something that you understand when you measure it. The problem I have with this is a philosophical question. Maybe you can give me some feedback. Is that there are many instances where it's not well defined what information is. So, for instance, if I ask you how much information is there in a bookshelf full of books, you can define it in many different ways and give me many different answers. So if you have large books and small books, you can decide whether they are random or they are ordered, and then you assign some bits and you give me some information. But if you order them by alphabetical order, then you have another type of information. And if you actually look at the content of the books, then you have much more information. So when I do the procedure of maybe ordering a bookshelf by alphabetical order, I'm sure that my brain is dissipating some heat, and when I forget it, this information is lost. But how much information is lost? It looks to me that depends on how you define the information of this bookshelf, but the energy that is dissipated in my brain doesn't seem to be something related to how I define the amount of information. Do you see my point?
Yeah.
That sometimes it's very difficult. I mean, it's like not an intrinsic property, the information. Probably in very simple physical processes, that the ones that you're studying, it's well defined. But in general, information, I think that it's not defining any objective way that you can really so easily translate to entropy or to energy. I don't know what is your impression of that?
Absolutely, you are right. I mean, I agree completely with you. And I don't know, because if we talk about information, then it comes to our mind and Shannon information. But of course, Shannon information is not everything. There are other types of information. There is Fisher information. There is information is a field in which we still do not understand what is the real we don't understand yet what is a real quantity. We should actually answer your question. So your question is well posed. When you have many books, you have books in the shelf, they are ordered in a way or another. What is information? Okay, so I can only tell you what I think could answer your question, and this is something I'm now behind by combining experiment and theory. I think that in the same way you saw in this example of the Szilard engine, okay, the work that I can extract depends on the observations that I made on the particle in the vessel. Every observation is a bit, can be 0, 1, and therefore the amount of the work that it gives me, this sequence of observations, the mean value of the work is given by this expression. And this is very simple thing that you know, that's the work that actually a series of sequence of observations can give you, okay, in average. Now, it turns out that this is equal to the energy that you need to erase those bits, okay? And this is a Shannon information contained in this sequence. So my statement is that the information, whatever it is, in the books, in the shelf, if they are ordered, not ordered, or the information that I keep in my brain, in my neurons, by the firing state of my neurons, whatever it is, my statement, that would be my statement, okay, is that the information content, whatever you can define as information content, okay, is the energy that you need to produce such a state. So actually, in my mind, or in my understanding, information is a work. It is a work that you need to go from one state to another. So the information that I gain when I study is a work that I have to do to study this topic. The information that I have to do I don't know, to read a book. The information that I learn when I read a book is the work that I have to do to learn that book. And of course, it's a relative quantity. What you said is true. One thing is information content, but it's really a content. It's like the internal energy, which is something that you define from the configurations, or is a relative quantity to another state. I think that what you said, that is a relative quantity, is the right answer. Okay, so I can only tell you that two things, that I think information is a relative quantity, I agree with you, but I think information as a relative quantity is a free energy difference. That's my second statement. But I don't know.
Okay, this is a very long debate, so we can discuss it.
Yeah, but it's a relevant, it's a very cute one. And
very
single molecule is that you have, you see, the good thing of single molecule is that you have these questions, you can answer them on a methodical basis, one step after another, because it's a simple system. You know, follow the state and follow the state. That's a nice thing of these experiments that are simple, actually are very simple, because you don't have, molecules have two states. They don't have an infinite number of states where you don't see nothing. You can coarse grain everything and reduce your problem to a very low number of degrees of freedom, which is basically the same that happens with the experiments that Pietro does with beads. Okay, our beads, micron-sized beads, have a low number of degrees of freedoms. Of course, in cells, as you do, for example, Jaume, it's much more complicated because cells are amazingly complex.
Yeah.
Okay.
Let me make a short comment just to finish. My impression is that one still requires a much more sophisticated way to define information. Because, for instance, if you let's consider a book like the Quixote, and you take all the string of characters from the first to the last, this contains certain information. Put exactly the same, the characters in the particular order has some information, right? If you put the same characters in a different order that make no sense completely, they have exactly the same information. It's a string of 10 to the something characters, and to specify that sequence, you need exactly the same number of bits that if to specify the sequence of the Quixote, right? If it's a random one. But obviously there are complicated correlations in the characters that define the Quixote that give us much more information, right, than just a random sequence, right? So if you take just the bits contained in the string of characters, that's a very poor description of the information that is contained in the Quixote. You see what I mean? So
I
there
absolutely
are
agree.
many levels of correlations and things that involve. It's not just bits. That's what I'm trying to say.
I completely agree with you. And actually, the energy you need to write the Quixote is much higher than the energy of a student that writes randomly letters in an exam, you know? I completely agree. That's the reason why I say that I think information is energy.
Okay.
And I won't say anything else.
You always fulfill second law, basically. Yeah.
Well, I want to say something else, yeah, because can I speak in Catalan or because we are 5 people here, right? No.
Not so much, but okay.
No. Bueno, estem entre amics. Vaig a dir una cosa. Si penseu en la segona llei és l'única que és una desigualtat en física. Jo sempre t'ho he dit, això, Jaume.
Sí.
Falta un terme allà. There is a fucking term missing there. Perdó que ho digui així, però la segona llei és la més estranya de totes. Totes les lleis de la física són lleis de conservació. És l'única que no és una llei de conservació. Falta alguna cosa.
Però d'altra banda, la més fort.
I
D'altra banda, què has dit?
O sigui, és la més fort de tot. És la que...
La più bella.
Bueno,
Bella.
però a part que la pots escriure com una desigualtat. Però la pots escriure com una desigualtat, però pots escriure com una igualtat, perquè si tu escrius la relació d'equips diferencial que combina primer i segon principi és una igualtat diferencial en què hi ha entropia i energia i...
És un balanç energètic, Jaume, és una llei de conservació de l'energia. Les igualtats són lleis de conservació d'energia. TDS, tot allò és Gibbs, Duhem, tot. La segona llei és el principi de claustres.
No, però està continguda, la segona llei està continguda en una TDS. La segona llei està continguda en les TDS. Tu pots escriure la segona llei amb una igualtat. La pots escriure, vull dir, això...
Bueno, la igualtat de Jarzynski et dona la segona llei. Un diria això.
El que vull dir és que... De fet, el caràcter de la desigualtat està lligat al trencament de la fletxa del temps. O sigui, és un trencament de simetria, en el fons, o sigui, totes les lleis, el mateix statement que estàs dient és totes les lleis de la física són reversibles en el temps, excepte la segona llei. O sigui, totes no tenen fletxa en el temps, per això parlem de conservació.
L'equació Schrödinger és reversible, és una primera derivada de la funció.
Per això, totes les lleis, les equacions de Maxwell, l'equació de Schrödinger, totes les lleis de la física són reversibles en el temps, excepte la segona llei. Vull dir, que ho pots plantejar com, més que una cosa de conservació, també és la simetria temporal. O sigui, és l'única llei que trenca la simetria temporal.
Bueno, hem de parlar junts, ja parlarem. No, perquè, clar, tenim coses entre mans tu i jo amb la conferència, etc. Ja parlarem.
Vale, vinga.
Gràcies, nois.
Muchas gracias, Felix. Thank you very much, Felix.
Noies, gràcies.
Thank you, everyone, for coming. I'll see you in the next one.
Vale.
Bye.
Merci, adeu. Gràcies, Felix. Adeu.
Adéu, gràcies.
Gràcies.
Arrivederci.
Gràcies, adéu.